Good evening. Thank you, John, for organizing it, and thank you all for joining. I'm assistant professor in Department of Radiology and works in the MEC core in the group of Matti Hamlainen. I joined Martinos Center in 2009 as a research assistant, thinking I will stay here for about an year, but it's been 12 years, and I am still here. Okay. And it has been an incredible journey. In 2019, I took an year off to work in the industry to see how do we do research in a more applied environment. I joined back the Martinos Center in April 2020. Today, I will be presenting our new paper, which shows how different brain regions orchestrate and communicate with each other to detect a moving object by a moving observer. This problem is known as SLAM in robotics and autonomous systems. I will present what SLAM is, why it is important, how we solve in engineering, and finally, I will present the neural dynamics behind the SLAM. Okay, so what is SLAM and why it is important? Simultaneous localization and mapping SLAM is a classical problem in computational geometry with applications in robotics and autonomous systems to simultaneously localize while moving, but also tracks other moving targets. In other words, when an object is moving in a complex environment in which other objects are also moving, to successfully navigate, it needs to track its own position, but also other objects moving around it with respect to its own movement. SLAM in an autonomous systems is usually estimated by fusing proprioceptive sensors like accelerometer, gyroscope, magnetometers, which measures the local position and orientation, and exteroceptive sensors like traditional RGB cameras or cameras with a depth channel RGB-D cameras, lidars, and lasers. These sensors measure movement with respect to the outside world. In some cases, especially when we have rovers in outside world in Mars and other, proprioceptive sensors are not available in an autonomous systems. And SLAM need to be estimated using only the exteroceptive sensors. In this case, SLAM is known as visual SLAM or VSLAM. Many algorithms have been developed to compute SLAM, from traditional control engineering, Kalman filtering-based approaches to a more recent deep learning approaches. However, SLAM remain a very challenging problem in computational geometry, especially the visual SLAM remains very challenging. As you can see in this video, I am showing this is from the 2006 RoboCup final, in which two teams from the fully autonomous robotics team compete against each other. Unlike robotics that we've seen on our Facebook or on Twitter feeds from Boston Dynamics, which are semi-autonomous systems which are controlled by the humans, there is no human input once the game has been started. As you can see, this is the current state of a fully autonomous robotic systems. In contrast, our brain solves SLAM effortlessly by integrating information across sensory systems like visual, auditory, proprioception, and vestibular systems. However, how does the brain resolve this problem and fuse information across various sensory systems remain largely unknown. In the last few years, there are multiple papers that has tried to find out the brain dynamics behind the SLAM. However, brain dynamics behind the SLAM, when SLAM is estimated only using the visual input through our visual sensory system, has not been explained before. In our recent paper in Progress in Neurobiology, using machine learning on MEG data, we decoded the neural dynamics behind the visual SLAM. We show how different brain regions orchestrate and communicate with each other to detect a moving object by a moving observer. I will now present the experiment that we used for the SLAM. As you all know, due to the limitations we have in the current brain imaging methods, like MEG and fMRI, subject remains to be static. So we develop a task that induces a perception of a forward self-motion. This task was validated in behavioral studies published earlier by our group. This task consists of an initial period of 300 milliseconds in which a red dot, a fixation dot appear on the screen. This is followed by a period of 200 milliseconds in which 9 spheres started to appear on the screen, and they achieved their maximum contrast by 200 milliseconds. After this, they remain static for about 1,000 milliseconds. Then the period of the motion started, and they start moving except one sphere. And this motion lasted for about 1,000 milliseconds, in which 8 out of 9 spheres move, inducing in an observer a perception of the forward self-motion, while one of this sphere moves either away or toward the observer with a different speed. This is followed by a period of 250 milliseconds of blank period. After this, subject is allowed to response, which is a 4 alternate force choice task in which subjects needs to identify which of these 4 options, one of which of this number corresponds to the sphere that is actually moving. And the response time period that we allow is about 1450 milliseconds. And after this response, that when they have detected which sphere was actually moving, we convert this response to a binary response. Either the response is correct or an incorrect. We collect 160 trials of this task across our participants, and we collected the data from 9 healthy volunteers. MEG data was collected during this whole task at a millisecond resolution. We use the data from 204 planar gradiometer, as we see over here, to perform a multivariate pattern analysis using support vector classifier in this 204 dimensional space to discriminate which brain regions corresponds to the correct versus incorrect responses. To discriminate correct versus incorrect responses, SVC, support vector classifier, learn a linear decision rule where the weights vectors and the threshold define an hyperplane. And this hyperplane divides the data into 2 classes, either correct or an incorrect. Just for visualization, we show 2 channels over here, but this classification has been performed in a high dimensional 204 dimensional space, and our plane is a hyperplane. To train and test the SVC, we use this 4-fold cross-validation in each subject, and the average prediction accuracy was computed across participants for each time point to characterize the classification performance from the motion onset from here to the end of the response. This slide presents the brain dynamics that leads to the correct response as inferred by the support vector classifier. This figure consists of 3 panels, as you can see. The top panel shows the task, which I have explained previously. Middle panel shows the accuracy of the SVC from the onset of the motion to the response. And the bottom panel shows which brain regions and at these particular times corresponds to maximum discrimination between correct versus incorrect responses. As you can see in the middle panel, at first, SVC briefly passed the threshold level at 150 milliseconds and then went below the chance level. After this, SVC reliably classified the trial with correct responses after 400 milliseconds from the motion onset, and reaching the highest accuracy between 1000 to 1200 milliseconds. These SVC patterns have distinctive spatial pattern at different time points. In the bottom panel, as you can see, that we're showing this at 4 different time points on the surface of the cortex. As you can see that at 600 milliseconds, when it started to continuously increase the chance level, that we have activation in the superior temporal circus, SPL corresponding to the superior parietal lobe, and the post-central circus. At about 1000 milliseconds from the start of which marks the end of the stimulus motion, this activity expanded extensively across the cortex. And we have activity in dorsal, lateral, prefrontal cortex, insula, inferior frontal gyrus, motion sensitive areas, as well as some visual areas. At about 1400 milliseconds, the participants are already in 150 milliseconds within the response period. And we see activation in the frontal eye field regions, inferior frontal gyrus, while the left hemisphere also shows some activities in the temporal and parietal occipital cortex. And about 1800 milliseconds, when majority of the participant has already responded, we see activation in medial orbital frontal cortex, superior frontal gyrus, and supplementary motor areas. We also look at how different brain regions exchange information between each other to orchestrate the SLAM. We use dynamic Granger causality in the source space to determine the time-varying functional connectivity between regions of interest involved in visual and visual motion detection, as shown over here. In each time window, the signal were fitted with an autoregressive model to compute the Granger causality. As you can see, there are spheres over here, the red sphere representing that it is acting like a source and sending information, while the blue sources that were shown over here are receiving information. And size of the source tells about how much the information is being sent or how much information has been received. As we can see in this figure, that different brain regions at different time points orchestrate and communicate information to achieve the visual SLAM. So we show that the multivariate pattern analysis using support vector classifier can decode the neural dynamics of the SLAM. Our brains solve SLAM by orchestrating and communicating information between visual, motion sensitive, and frontoparietal areas. And importantly, these findings not only help us to understand the brain functioning better, but might also provide us novel ways to solve SLAM in robotics using brain-inspired approaches. Thank you all. And this work is a synergy between BU Brain and Vision Research Laboratory led by Dr. Lucia Vaina and MEC Core at Martinos Center led by Dr. Matti Hämäläinen. Thank you to our joint first author, Natalia, Adone, Abhi, and thank you, Kunjan, for doing the Granger causality. Thank you all.